Welcome everybody to the webinar. We're just waiting for a few more participants to trickle in, and then we will begin the webinar in less than a minute. Okay, we're going to go ahead and get started. Welcome to the Research Coordination Network webinar on SARS-CoV-2 in wastewater. This is an NSF sponsored RCN, and we've been doing these webinars every 2 weeks. If you're on this webinar and you're not on the current listserv, please send me or Jill McCleary a note and we'll add you. Today, we're happy to have 2 people presenting. We have Dr. Nadine Kotlars from North Carolina State University, a postdoc there, who will be speaking first. And then our second speaker is Zach San, a PhD student from CU New York. And the way these webinars work is that the webinar participants aren't given speaking abilities, so all the questions go into the Q&A. Don't put them into the chat, put them into the Q&A. And then once someone enters a question in the Q&A, you can upvote it and it can rise to the top, and a moderator from the panel up here, which consists of the speakers and the PIs on the RCN, will ask the questions of the speaker. And if necessary, we can allow speaking access to a participant. So again, this is a webinar for what we're calling junior researchers. That includes assistant professors, postdocs, and PhD students. And if you know someone who would like the opportunity to present at the webinar, also please let us know. And we're here to really share information and also celebrate all of the hard work and service that our speakers and attendees have devoted to the pandemic, which we all hope ends very soon. So with no further ado, I'm going to pass the torch to Nadine, who will be our first speaker.
Hi, everyone. Thanks for this chance to tell you about our work. My name is Nadine Kotlars. I'm a postdoc at NC State University, and I'm presenting on behalf of our team, Angela Harris, Stacy Reckling, Helena Matasheva, Jane Hoppin, and Francis De Los Reyes. So we're in North Carolina, in central North Carolina, and we're working closely with the Raleigh sewer shed. I'm just showing you a map of the sewer shed, and the utility is located in the southeast corner. The sewer shed serves about 580,000 residents. We've been collecting samples from this sewer shed 2 to 3 times per week since April 10th. We're collecting it at 4 locations. We collect influent and primary solids from the head of the plant. We've been collecting grab samples of wastewater from 2 neighborhoods at their wet wells, and we've been collecting composite samples of wastewater immediately downstream of a local hospital that has COVID-19 patients. Today, I'm just going to focus on the analysis of the solid samples. So in thinking about trends tracking and using solids data for trends tracking, there are some possible strengths of monitoring solids. And so solids are the primary clarifier at the utility, serves to concentrate some of the signal in the influent through that concentration in the primary clarifier. So solids have potentially a higher frequency of detection than what you would see in influent, and this has been reported by some other teams. And so this means that you may be able to analyze solids using a direct extraction approach and avoiding a in-lab concentration step, which was a real consideration for us in the beginning because those in-lab concentration steps were triggering some additional biosafety requirements. And so what's nice about solids is that the signal may be higher there, so you may be able to analyze with a direct extraction approach. There are some possible limitations of solids, so that retention in the primary clarifier can cut into any lead time that you might see in the solids data relative to case data. There may be greater inhibition in a solid sample than in an influence sample, and solids monitoring is not really feasible for neighborhood level monitoring. So let's talk about the solids that we've been analyzing in Raleigh. So we've been collecting primary solids from a sludge pump, from the primary clarifier, and we also considered collecting our solid sample from the day tanks, but we wanted to reduce the retention time as much as possible so that any possible decay that could be happening would be reduced. So we decided to collect samples from the sludge pump. And when we're talking about Raleigh solids, what's important to realize is that at this utility, secondary solids are recycled to the head of the plant. And so the solids we're sampling from the primary clarifiers are actually a combination of primary and secondary solids. And so I'll talk more about that a bit later. So when we're analyzing solid samples, this is the approach that we're taking. We collect the samples and in lab dose them with a bovine coronavirus to serve as an internal standard. The samples are stored then at -80. We thaw the samples on ice and are extracting directly from 2.5 mils of solids using a QIAGEN RNA-Z Power Soil Total RNA kit, which has been used by others to analyze for SARS-CoV-2 RNA in solids. This Power Soil approach involves a relatively aggressive bead beating step in the presence of lysis buffer and phenol, and it is not a kit that can be easily automated, but it has resulted in good purity RNA from the solid samples that we've been working with. So we measure each sample, its total solids concentration and the total RNA concentration. And then we analyze for SARS-CoV-2 N1 and N2 targets using digital droplet RT-PCR, and we're duplexing those N1 and N2 and merging duplicate reactions. So we're effectively screening 0.5 ml of sludge in this pipeline every time we do a sample analysis. And we've so far analyzed samples from April 10th to October 17th. So just looking in on an example of what we're seeing in our solids, what we see, this is a 2D plot from a ddPCR, and so we tend to see more single positive droplets than we do double positive droplets for either single positive for N1, single positive for N2, and then fewer but some double positive for N1 and N2. And we are in parallel testing a SARS-CoV-2 synthetic RNA standard, and that exhibits a similar droplet pattern. When we do spike recovery of this synthetic RNA into a solids matrix, we see good recovery from the samples. And with our internal standard, we're seeing about 2% recovery from the solids and closer to 4% when that freezing step is not there. In solids, we have a moderately good correlation between the total solids concentration and the total RNA concentration. And so we normalize our solids results by total solids and report gene copies per gram solids. So this is a plot showing you our N1 and N2 data over the course of the sampling campaign. And it's in gene copies per gram total solids. And what we've seen is that using the direct extraction approach, nearly all of the samples that we've analyzed have been positive for N1 and N2. We've also tested 2 North Carolina solid samples that were collected before the pandemic and those were both negative for N1 and N2. There's a strong correlation between N1 and N2 in our data. So as we move through the analysis, we really just talk about N1 because these two are so highly correlated. So thinking about the sensitivity of this approach, the first lab confirmed case of COVID-19 in Wake County, which is the county that houses our sewer shed, was March 2nd. And then if you fast forward about a month, we started collecting samples on April 10th. And at that time, there had been about 200, we estimate about 200 cases in the sewer shed at that time, cumulative cases. And so, we're seeing detectable levels of CoV-2 RNA in the solids on this day, but it is getting close to the limit of detection. We're starting to see single-digit positive droplets. And so, this method has worked well for this time series, but if we had wanted to increase sensitivity, we would have needed to consider a larger sample volume for direct extraction or some concentration step. So over the time series that we collected samples, this direct extraction approach is working well for trends tracking, but had we gone back further, we probably would have needed a potentially more sensitive approach. So recall that I told you our primary solids are not really just primary solids, they also include secondary solids. And this is not really typical wastewater treatment. The secondary solids are co-settled in the primary clarifiers at our utility to increase solids concentrations there to help with settling. But this is not something that's typically done across utilities. And so we were interested in looking at whether there was any detectable SARS-CoV-2 RNA in the secondary solids to help us interpret some of the results we're getting from the primary clarifiers. We sampled primary and secondary solids over 4 consecutive days at this plant. And so I'm just showing you N1 copies per gram total solids in primary solids and secondary solids over these 4 days. And what we see is that SARS-CoV-2 RNA was detectable on all 4 days in the secondary solids, and that at least on 2 of the days, the levels were comparable with primary solids. The current CDC guidance is not to use secondary solids for wastewater surveillance, but for utilities that don't have primary clarification and are interested in doing solids analysis, it would be helpful if there were more data on the presence of SARS-CoV-2 RNA in secondary solids. So we've talked about the wastewater results, and now thinking about how these wastewater results relate to health data case trends. We've been talking closely with public health stakeholders, and some of the input we've heard is that individually public health officials realize the benefit of wastewater surveillance, whether or not it agrees with case data. And even though that's true, a majority of them want to see that relationship anyway, whether it agrees or doesn't agree. And demonstrating good agreement is going to be helpful if the wastewater data are to be actionable. So it's important to think, I think, carefully about how we test this relationship between the wastewater data and the case data. So ideally what you'd have is individual level geocoded test results for a COVID-19 positive case that you could say, confidently, given its geocoded location, it's located within the sewer shed boundary. But geocoded data are protected health information. You need to often submit data requests to your health departments to access these data. And you also importantly have to have the proper data security infrastructure to protect those data. And so what we've been dealing with at NC State is that because we do not have a medical school, we did not have the infrastructure to properly meet the data security requirements that were imposed by the health department that was wanting to share the data with us. And so we're working on resolving that, but it led us to ask questions about what's possible with publicly available data. So daily zip code time series data are publicly available, and you can use them to try to approximate cases in your sewer shed. And there are challenges there because, of course, zip code boundaries don't align perfectly with sewer shed boundaries. So the question becomes, how do you aggregate zip code level data to approximate cases in a sewer shed? And so we've looked at this and tested 3 different approaches to this. So the first thing you could do is just sum all the cases in all the zip codes that intersect the sewer shed boundary to any degree. You could take an area weighted approach where you weigh each zip code's case count by the proportion of area in the zip code that overlaps the sewer shed boundary. You could take a population weighted approach where you weigh each zip code by the proportion of population in that zip code that overlaps the sewer shed boundary. And so let me be more clear with an example. So this is, again, a map of the Raleigh sewer shed. The yellow is the sewer shed boundary. The blue lines are delineating census blocks, and census block population estimates are available on census.gov. They're publicly available data. And the black lines are showing you zip code boundaries. So if we focus in on one zip code that is only partially located within the sewer shed boundary, we can get estimates of how many people live in each of these census blocks. And so for this zip code, if we were to adjust by area, we would say that the overlapping area of this zip code in the sewer shed boundary is 0.21. That's the adjustment factor that we would use taking an area weighted approach. But if you took a population weighted approach, because there's more densely populated pockets in this zip code, you would actually weight the cases from this zip code higher if you took a population weighted approach. And so this just illustrates the differences between these 2 approaches. And so we've tested all 3 approaches in the Raleigh sewer shed. So I'm just showing you the case estimates for the sewer shed using those 3 different approaches with publicly available data from early May to January 2021. And so one of the first things that we can see is that, so the red line represents all of the zip codes touching the sewer shed boundary. The green line is the population weighted approach, and the blue line is the area weighted approach. And so you can see that if we took an area weighted approach in Raleigh, we would be undercounting the number of cases in the sewer shed. A population weighted approach includes corrects for the fact that population is not uniformly spread across those zip codes. The other thing that I think stands out is that no matter what approach you use here, the trends are pretty similar. And so this is the case in Raleigh, where we have a couple of large, entirely located in the sewer shed zip codes that are densely populated and seem to be driving those case trends for the Raleigh sewer shed. It may not be the case that these 3 approaches agree if you have a sewer shed in another city that has sort of different population densities and different zip codes overlaying with that sewer shed. So some important considerations in how well these approaches agree is how many of the zip codes are geographically entirely located within the sewer shed boundary. That's going to matter in how well they agree. And for the zip codes that are partially located, how uniform is the population density across that zip code? So just something to consider if you are not able to access individual level geocoded test results and looking to work with some public data to approximate your sewer shed, approximate cases in your sewer shed. So we took the population weighted approach because it seemed the most defensible, and we compared our wastewater data with the new cases rolling average using that population weighted approach, and Pearson's correlation coefficient between the 2 rolling averages was 0.77, suggesting that there's a strong and good correlation between the wastewater data and the case data. We were interested to see whether there was any difference in that agreement if you look at earlier in the time series or later in the time series we thought there might be poorer agreement earlier because there was less testing done earlier. But what we saw was better agreement early on in the time series than compared to the second half. So we just divided the data set in half and looked at the correlation coefficient over those two halves. You know, possible reasons for why the later time series is not as good agreement could be related to some of the operational changes we've learned happened at our utility. And so we're still sort of teasing this out. It may also not be related to operational changes. I've just heard anecdotally from other teams that they are seeing better agreement early on in the pandemic. So we've sort of looked at, you know, the agreement over time, but we also wanted to think about whether the agreement changes if you restrict the analysis spatially. And so this is just travel time estimates across the Raleigh sewer shed. And so you can see that for the most part, wastewater travel time is between 3 and 12-ish, sometimes 15 hours. So not especially long travel times. And if we look at the half-lives that have been reported for N1 in the literature, it's possible that there may be some decay happening, but it's also possible that there isn't. So we wanted to restrict this analysis to the zip code that was closest to the utility and had the shortest travel time. So when we look at the Pearson correlation for the cases in this zip code relative to the wastewater solids data, we still see good agreement, but we don't see better agreement than we did across the sewer shed overall. So I wanted to describe some ongoing work we have where we're looking at wastewater trends, not just against case trends, but we're becoming increasingly interested in comparing them against syndromic case trends. So COVID-like illness is defined as a positive case based on symptoms and does not require a lab-confirmed test to be counted as a positive. And these cases are identified through questions that are asked at emergency departments. So in North Carolina, we have extensive syndromic surveillance that's being leveraged for flu, but it was also used to track COVID-like illness through the pandemic. And we have several hospitals that are participating to report these COVID-like cases to the state. And so we've been working on accessing the syndromic data to take a look at how well the syndromic data agrees against the wastewater data. Some of the pros of looking at syndromic data include that it's testing independent and that it could capture populations that are not captured by the traditional testing approach. So the cons would be that it relies on individuals who are seeking care at emergency departments, and that like many things in the beginning of the pandemic, the emergency department visits were lower than they were later in the pandemic. So in summary, we have detected SARS-CoV-2 RNA in nearly all of our primary solid samples using a direct extraction approach. We approximated the sewer shed cases using publicly available data in 3 different ways and found similar trends in Raleigh. SARS-CoV-2 RNA in solids is strongly and positively correlated with cases. We see better agreement early in time, and when we restrict our analysis to a zip code close to the plant, we don't see an improvement in that agreement. And we are working on evaluating relationships between wastewater and syndromic cases. I'd like to acknowledge our team, our sources of funding, and if you are also working on communicating wastewater results to public health stakeholders, we'd love to talk with you more because we're actively working on figuring out how to do this well. And so thanks for your time.
Thanks, Nadine. That was a great presentation. Just a reminder to everyone, if you have any questions, please put them into the Q&A box. And you can also upvote questions that you want answered. And so we'll kind of work our way through some of these ones that have already come in. So the first question is a methods question. I think you mentioned you ran the power soil extraction kit. Did you ever try the power microbiome kit and compare against that?
We haven't, but we plan to because we've had supply issues with the power soil kit. And so the power microbiome is on our list of alternative approaches to consider.
Great, thanks. And then next question, related to the solids data. So this is sort of about applying that data to a health situation. How do you think solids SARS-CoV-2 data would be best utilized given current levels of high testing, clinical testing, and shorter lead time, potentially with solids data?
I think that's a good question, and one that we want to continue to think about. And we collected influent at the same time that we collected solids. So as we analyze our influent, we're going to be looking at sort of the relative advantages of solids to influent analysis. But I think it's a good question to ask whether the methodological advantages of a solids analysis outweigh the potential delay in the timing. So something that we are working on and thinking about.
Thanks. So now a question related to travel times in the sewer shed. Did you have information about the pipe network, or did you have to assume an average diameter? And a related question was about whether or not sewer shed data were publicly available, or if you had to get that from the utilities. Maybe you could describe a little bit of working with that sewer shed data.
So my understanding is that we do have extensive pipe network data for Raleigh. We are also working with other utilities that don't have as good a data, but for Raleigh, we did have good pipe data that was used to calculate those velocities.
And then just that follow-up, was the sewer shed data you received from the utility then that wasn't publicly available?
So the polygon, the shape polygon was received from the utility.
Great. And let's see. We have, you mentioned that you've discussed with some other teams that are seeing better agreement with their data early in the pandemic. Do you have any guesses why that is? I know it was sort of contrary to your hypothesis.
I don't have any well-developed guesses. There's been some discussion that it has to do with fecal shedding, but I don't know exactly why, and I don't know. I think the first thing we'll do is look at within our system if there's any clear reasons that would drive that kind of agreement. But then across systems, I'm not sure.
I think that's something that we'll all be kind of looking at soon. And then I think we have time for maybe just one more question. Have you considered mutation of the N gene or asymptomatic infections and how those might contribute to your model analysis?
So we are interested, our team is interested in looking for variants on that may be present in the wastewater, and we're developing methods that will allow us to do that. So with respect to so the wastewater in theory is capturing asymptomatic shedders, whereas the health data are likely not capturing those individuals. So I think I'm not totally clear on how the question, but I think what we'd like to do is be able to sort of leverage the wastewater trends to understand better these two surveillance systems in North Carolina, one that relies on testing and one that relies on syndromic surveillance. But in both cases, you're relying on people who seek healthcare for one reason or another. So, really the wastewater is it's capturing the asymptomatic fraction.
Yeah, absolutely. Great. I think for the sake of time, we'll switch things over. Thanks so much, Nadine. That was a great presentation. And before I turn things over to our next presenter, Zach, I wanted to just briefly mention, for those who may not have heard, just a quick reminder that the RCN is planning a workshop on standardizing data reporting. for wastewater SARS-CoV-2 data, and nominations for that workshop are closing tomorrow. So I'm going to put a link in the chat that will take you to more details and the nomination form for anyone who's interested. And with that, I'd like to turn it over to our next presenter, Zach Son from City University of New York. Zach, you can go ahead and share your screen and start whenever you're ready.
Thank you very much for the nomination to speak in today's webinar. My name is Zach, and I'm a graduate research assistant in Dennehy's lab from CUNY, Queens College. Today, I want to talk to you about our project on the SARS-CoV-2 surveillance and how we optimize our protocol to detect them in the wastewater from the New York City. And this is a collaborative project between the CUNY team led by Dr. John Dennehy from Queens College and Dr. Monica Trujillo from Queensboro Community College. Also the New York City Department of Environmental Protection and NYU School of Engineering, and then the New School. So in the previous seminar, Catherine, if you do remember, for Catherine from NYU had talked about how we monitor the SARS-CoV-2 and show some of the trends that we're seeing here in the New York City. But today I'm gonna discuss on how we had developed and optimized our protocol to quantify and maximize the SARS-CoV-2 recovery from the wastewater. With that, as we all know, there are challenges when it comes to detecting the SARS-CoV-2 from wastewater, either for the environmental surveillance or studying for the academic research. These challenges mainly come from not having the standardized protocol. So before we start, we need to carefully assess on the method that works best from all the other existing methods. The challenges may also arise from not having enough fundings, okay? So here we assess and develop the method not only works well to quantify the SARS-CoV-2 RNA in wastewater, but also at the same time cost effective. So in general, sample processing for measuring the SARS-CoV-2 RNA in wastewater involves sample preparation and then sample concentration and RNA extraction and detection and quantification. But it's important, the method that we selected at each step must be carefully evaluated for the better detection and quantification. So let's start with the sample preparation. When we prepare the sample, we ask ourselves whether the wastewater should be pasteurized or not. Yes, but we think it should be pasteurized for two reasons. One is for the safety reason. We pasteurize to reduce the chance of infection, not just from the SARS-CoV-2, but also from the other waterborne pathogens. So two, we made a comparison between the pasteurized and an unpasteurized, and we found that pasteurization helps recover more SARS-CoV-2 RNA. So based on that, we chose to pasteurize our sample. So after pasteurization, should the wastewater be filtered to remove these solids? So there's a mixed report over whether the matrix should be removed or not, because the viral particle may bind to the solid matrix. But on the other hand, the solid matrix may contain the non-target nucleic acid from other microorganisms. So practically speaking, solid matrix makes it difficult to process our sample. So for this reason, we remove them by doing the low speed centrifugation, and then we filter the supernatant. So how does the filtration affect the SARS-CoV-2 recovery then? And so we compare the filter and the unfiltered wastewater, and we found that although we lose some of the SARS-CoV-2 by filtration, the effect of the filtration is very minimal. But the filtration can offer a benefits by removing the solid impurity for the downstream processing, such as like RNA extraction and then qPCR. So based on that, we chose to filter our samples. This data for the filtration was presented in the previous webinar by Catherine from NYU. So after filtration, should the virus be concentrated? So if so, what's the best mean of the concentration? So if variants are highly diluted in the wastewater, so we need to be, they have to be concentrated before extraction for RNA. So there are two different approaches. So one approach is to concentrate the viral particle by binding and precipitating them with this substrate using the aluminum chloride and skim milk or the polyethylene glycol, PEG method. Another approach is to use ultracentrifugation. So we evaluated all four methods and we found that PEG, polyethylene glycol, PEG method, to be the best because it gives the highest SARS-CoV-2 signal when we run the qPCR compared to the other concentration methods. So after concentrating the viral particle, we do the RNA extraction. So this is a very important step. It is crucial to have a good technique in order for us to obtain a high quality RNA. We need to carefully design and select the protocol that can produce a highly purified RNA, high yield, also that would allow us to detect and quantify SARS-CoV-2 without any inhibition in the downstream processing. So for the RNA extraction, there are two basic strategies that we can use. We can use the commercially available column-based kits, okay? And then the trizol, okay? So there's a trizol phenochloroform methods. For us, we use a trizol phenochloroform method as this method has worked really well in our hands and has been employed and recommended by the other groups as well. So finally, after we obtain the RNA, we can quantify them using a qPCR. So for us, we found that targeting the N1 gene gives a much better signal than the other gene targets. So we use the N1 to detect the SARS-CoV-2 from all our wastewater sample. There are multiple testing methods and a laboratory workflow that are used to quantify the SARS-CoV-2 in wastewater. But here, based on what we had tested, we created the workflow on how we process our wastewater sample to detect the SARS-CoV-2. We begin by pasteurizing our wastewater sample at 60 degrees Celsius for 1 hour. And then we separate out the solid and other matrix. Then we filter the supernatant, and then we add the PEG and sodium chloride into the filtrate to precipitate the viral particle. Then we extract the RNA from the precipitated variants. And finally, we use the extracted RNA to quantify using a one-step RT-PCR method. So in the previous webinar, Catherine from NYU did such a great, amazing job presenting the data on the effect of pasteurization and solid separation and filtration on the SARS-CoV-2 recovery. Today, I want to continue with the PEG precipitation, RNA extraction, and RT-qPCR, and discuss on how we tailor within each of these steps for the better quantification of SARS-CoV-2 from the wastewater sample. So also taking the cost into consideration. So one thing that we looked at to assess was the effect of storing the pasteurized wastewater on the SARS-CoV-2 recovery. We wanted to find out whether the sample needs to be processed immediately after pasteurization, or if it can be stored to be processed later. To find out, we process some wastewater sample immediately after the pasteurization and store the rest of the leftover for 96 hours, which is about 4 days at 4 degrees Celsius in the fridge. Our data indicates that there is the decline in the viral recovery across all wastewater treatment plant after we left and store the sample for 96 hours at 4 degrees Celsius, as you can see on the graph. For some of the wastewater treatment plants, there is a small decline, but for most of the wastewater treatment plant, it is pretty significant. So this result is kind of suggesting that storing the wastewater post-pasteurization sample has a detrimental effect on the recovery of the SARS-CoV-2 RNA. So is there a way that we can manage to pause at some point and store them without degrading the SARS-CoV-2 if you want to process them later? To answer the question, we left the sample stored in the polyethylene glycol, the PEG, and the sodium chloride to see whether leaving the sample in the PEG for longer time has any effect. So we have hypothesized that leaving the wastewater in the PEG will be more stable from degradation after removing the solids and most of the impurity from the wastewater, because PEG basically does nothing damage except to help precipitate the viral particles from the wastewater. So we made a comparison between the wastewater that was stored in the PEG and sodium chloride for 24 hours and 48 hours. So as you can see on the graph, so our data indicates that there is no decline in the viral recovery if we leave the wastewater in PEG in most of our sample that we tested. But in some sample, longer precipitation time even helps by increasing the number of viral copies. But just note that leaving the wastewater in the PEG longer than 48 hours does not maximize the viral recovery. This data suggests if we want to store the wastewater for the processing later, it is better to store that in the PEG since the variant seems to be more stable in the PEG than the wastewater itself. So another thing that we looked at to assess that we thought it might be helpful when we processing our sample is the use of safranin. So here on the slide, you can see here's a key step where we introduce safranin to optimize our protocol. And one thing that we noticed and found it difficult during the sample processing is the visibility of the pellet after centrifugation. So sometimes we can see the pellets, but most of the time the pellets are invisible. The visibility of the pellet is very important for maximizing the recovery. So to be able to visualize the pellets, we can use the help of safranin. So this helps maximize the recovery of the SARS-CoV-2 because by being able to visualize, you can make sure you dislodge and pick up all the pellets when you wash them with the Trisol or washing buffer if you use the kit. This is also to help us control our data for consistency, to be able to compare between sample. The inconsistency can come from not dislodging all the pellet from the wall of the tube after high-speed centrifugation. So that's why we developed this idea of using safranin to stain the pellet so that we can visualize and make sure the pellet is dislodged from the tube when we wash them with the Trisol or washing buffer to be processed later for the RNA extraction. So we made a comparison on the SARS-CoV-2 recovery between the sample that we processed without safranin, which is our control, and with 2% safranin. As you can see here in the picture, without safranin, there's no visible pellet. It's pretty empty. And by adding the 2% safranin, you can clearly see the pellet on the wall of the tube. So those tiny little dots on the tubes are the pellets that we can see them because of the safranin. And we noticed that the amount of shaking, vortexing that you need to do to dislodge these pellets varies from one sample to another. Some pellets are big, some are small, some wash off easily, some are really sticky. So here I want to play you a video to show how well the pellet sticks to the wall of the tube after centrifugation with the high speed for 2 hours. So you might have noticed that after we shake the tubes for the first couple of seconds, and all the pellet didn't got dislodged, the pellet got stay sticking in the tube. So after shaking the tube for a couple of seconds without safranin, you would probably assume that all the pellets are washed off from the tube, but no. Even after shaking the tube, the pellets stay sticking on the tube. So if you don't see the pellet, you might end up missing them. You might end up leaving the pellet in the tube. That will result you in the low recovery of this SARS-CoV-2 RNA. But by visualizing these pellets, we can make sure that all the pellets are completely washed off from the tube. Now we now know that adding safranin helps you visualize the pellet, but wouldn't it interfere with the downstream processing when we run the qPCR? So we made an assessment on that. We found that safranin didn't interfere with the down processing at all. And so therefore, like adding the safranin is a very good tool to help us visualize where the pellet is and make sure it is completely dislodged from the tube wall. So this proper dislodging then significantly increased the SARS-CoV-2 recovery. So please note that the number of the viral particle it's fixed in the wastewater sample. But the whole point of doing safranin is to see if we can get all those pellets from the tube. So in this case, safranin surely can help you effectively recover most of them to make sure you do not leave anything behind. So next, we also try to optimize our protocol during the RNA extraction step. We wanted to find out which extraction method is better. So we made a comparison between the RNA extraction using the trisol and then QIAGEN viral RNA mini kit. And we found that RNA extraction with the trisol result in significantly higher SARS-CoV-2 recovery than using the kit, as you can see here on the graph across all the samples. So the one with the trisol yields significantly higher RNA copies. So in addition to that, a trisol, not only it gives you more recovery comparing to the kit in terms of supply chain, like right now, everybody is buying the kit to extract the RNA, so we might not have enough for everybody. So trisol, compared to the kit, is less expensive. And so I would say this using the trisol is more cost effective. Finally, one step RT-qPCR, where we do the quantification of our extracted RNA. So at this step, there's a choice regarding which enzyme that we can use. Our goal is to have the tag enzyme that can do the high performance with affordable price for the routine. So we made a comparison between two different tag enzymes, tag pack Thermo Fisher and Takeda. The difference between these two enzymes is the cost. So Takeda is 25% less expensive than the tag pack, so it's more cost effective. But how do they differ in terms of their performance? So we did a test on both enzymes and I found that Takeda enzyme works as well or better than the tag path in most of the samples that we tested, as you can see here on the graph or data. And that's how we develop our protocol and tailor within each of the sample processing step to maximize the SARS-CoV-2 RNA from the wastewater sample. Also, we take the cost to process these sample into consideration. With that, I'd like to thank everybody from the CUNY team, NYU, the NU School, and the DEP for their contribution and their hard work to make this project and presentation possible. Thank you very much.
Great, thank you, Zach. So we only have one question right now in the Q&A, and I have a couple of others that I'm interested in asking, but if folks would like to add questions or upvote questions, please add them to the Q&A on the bottom of the Zoom screen. So the first question in the Q&A is, wouldn't the choice of viral concentration and RNA extraction affect whether pasteurization and subsequent filtration, or these steps that you were evaluating, improve or reduce the signal of the SARS genes? Essentially, I think asking how translational are some of these findings regarding the viral concentration and some of the methodological choices, depending on the upstream decisions that you and your team made.
So the question is asking whether the choice that we've made would improve the signal of the RNA.
No, I think that the question is asking about the fact that you found the best choices based upon your protocol, but essentially asking if you change the protocol. So for example, if you used a different viral concentration method, or a different RNA extraction method, do you believe that these findings regarding pasteurization and other processing steps would hold, or do you think that they're dependent really on the method pipeline that was used?
Oh yeah, so that's something that we can kind of test and compare. With the sometime for some other cases, the pasteurization could be better depending on how you pair up with the methods. So for some of the methods that we're still working on to see which ones are better. So this is what we have found so far. The one that I presented was what we found to be the best so far at the moment.
Great. All right, another question is, so you found that the Trizol method worked better, but was there any difference in inhibition between the Trizol and the QIAGEN kit?
In terms of inhibition, we did not see any inhibition from the Trizol and the kit as well. A lot of people think the kit would work better, but Trizol, it really depends on the operator. I know that processing the RNA extraction with the Trizol can be a bit difficult, and sometimes it can give you inhibitions. But the product of the Trizol, it really depends on the operator who operates this RNA extraction.
I guess as a follow-up to that, how did you assess inhibition for those?
The inhibition, how do we assess? I would probably, we actually did the dilution between the sample in the sample. For the RNA extraction, we just actually dilute the concentration of the RNA to see, if we get any signal.
All right, great. So another question is, and I'm going to add on to this one, what was the total processing time for this workflow? And then the part that I'm going to add is what was the throughput? So there's, sort of the two pieces a lot of us are thinking about was how much time does it take, but how many samples are you able to put through?
Okay, so for us, we process about 14 samples every week. So the part that takes a lot of time and takes longer is the pack precipitation time, and also centrifugation. So centrifugation, you have to centrifuge at the high speed for 2 hours. That also depends on the availability of the centrifuge. So if we have more centrifuge machines, then we can probably like centrifuge all the sample at the same time. So for overall, the total time that it takes for us to process is just, I would say 3 days.
Okay. And another question that was just upvoted. Do you have any data on the variability for this complete method? So you were assessing sort of comparison, but as well as sort of evaluating replicates and the variability through the processing steps.
Variability. So yeah, when we process a sample, we're not just only doing like one sample. We also include the duplicate and sometimes the triplicates to be able to see, some of the variability. Yeah, so that's what we did.
Great. And do you have any insight on those values?
The value, not on top of my head. I probably have to look into.
Yeah. Okay. Yeah, that's fine. And I guess the last question here, and just perfectly on time, is what was the number of samples compared to the extraction comparison? So basically, how many samples did you use to compare the Trizol versus the QIAGEN kit?
The Trizol and then QIAGEN kit, how many samples? We did a comparison across like 14 samples.
Okay.
Yeah.
Great. So with that, I think that's all the questions that were asked by the attendees, and it's almost perfectly on time. So thank you, Zach and Nadine, for great presentations, and thank you everybody for attending. And we'll look forward to seeing everybody at the next presentations in 2 weeks.
Thank you.